Okay, thank you very much, Monica, for the invitation. So in the next few minutes, actually, Monica asked me to discuss with you the example of a very good collaboration that we have with the CALGB in a particular clinical study in acute myeloid leukemia that I believe provides some insight on the opportunities, but also the challenges of this kind of collaboration. So these are my disclaimers. So we will be talking here about FLT3 mutation in acute myeloid leukemia. So FLT3 is a tyrosine kinase receptor that is frequently mutated in a subset of patients with AML. Approximately 20% of the patients have a mutation near the juxtamembrane domain of the receptor called the internal tandem duplication. These are the most frequent, and these are associated with a poor survival. In addition, a smaller subset of patients, about 10%, have activating mutation of the tyrosine kinase domain. And both of these mutations have attracted a lot of attention over the last 10 years, and this is how the story fits with a compound discovered by Novartis, Midostaurin, formerly known as PKC412. So this is an orally available multi-targeted tyrosine kinase inhibitor. Actually, initially, it was developed as an inhibitor of protein kinase C. And as a matter of fact, it is out of the collaboration with Dana-Farber that Dr. Benz mentioned that it was discovered in the early 2000s that this compound was also, at the time, the most potent inhibitor of FLT3 with inhibitory IC50s in the nanomolar range. And this prompted a number of investigations preclinically and clinically. Now, because of the nature of the compound, it also inhibits other targets that may be relevant to AML. That could be the case for KIT, that could be the case for PDGF receptor. Now, at the moment, our focus in the development of this drug is obviously on acute myeloid leukemia, but also in aggressive mastocytosis with data that have been presented at ASH last December. So how the story started for this collaboration between CALGB and Novartis. So Midostaurin had started actually the first clinical studies in 1995. At the time, that was an healthy volunteer study. And because of the initial targets that were considered for this compound, the compound had been investigated in numbers of indications in solid tumors as well as hematologic malignancies as a single agent and in combination. But the interest in AML came out very much in the mid-2000s, where activity was observed as a single agent in patients with mutated FLT3 AML, and that moved the compound into combination studies that also showed very good result with a very high rate of complete response. So this is where we were, more or less in the years 2006 or so. And we started to think about how to move the compound forward to the next step in a phase 3 study. At the time, the CALGB, who had already spent a lot of time doing some correlative science on notably the clinical significance or the prognostic significance of FLT3, had an interest to really do a phase 3 with a FLT3 inhibitor. The CALGB had a slot, so we started really to talk to each other. Now, from the very beginning, something was quite evident, that we were talking about a relatively rare condition, FLT3 mutated AML, and it became clear that a study in this setting would require a very large and complicated collaboration that could go beyond what the CALGB could deliver in itself. So there was also a sort of potential synergy with the infrastructure of Novartis at a global level to bring more cooperative groups or more collaboration to the study. So this is the design of the study. So it's a simple randomized phase 3, young patients with newly diagnosed AML who have either an ITD or a TKD mutation were randomized to receive a backbone chemotherapy with the classical 7+3 regimen with cytarabine and daunorubicin, followed by a consolidation. And then a maintenance treatment up to 1 year. And this was done either with midostaurin or with a placebo. It was a 1-to-1 randomization, and the primary endpoint for the study is overall survival. Obviously, responsibilities had to be well defined from the beginning. The CALGB was responsible for the writing of the protocol with input from other cooperative groups who participated to the study. The CALGB owns the database and is responsible for the organization of the DSMB reviews on a periodic manner. With regard to sponsorship, the sponsorship is with CALGB for North America, whereas outside of North America, that was Novartis. And importantly, I'll come back to that, for the FLT3 testing, it was a shared accountability between CALGB and the other cooperative groups. And obviously, down the road, if the study is successful, Novartis will have the accountability of filing that study to health authorities. So this is the setup. 13 cooperative groups are participating into that study in North America, in Europe, and in Australia. So you have some of the main cooperative groups in Europe, the EORTC, a couple of groups in Spain, a few groups in Germany, and you have also a group in Australia. The study started in July 2008. Now, key characteristics of that study that generated some complexity, actually, is the need to detect the mutation before the patient received the chemotherapy. And knowing the sort of clinical urgency of a diagnosis of AML, there was a very short period of time, a very short window of opportunity to really do this testing. And having this in mind, the decision was made initially to use 10 central laboratories in each of the main cooperative groups. So here are the laboratories who participated in this trial. Now, all these laboratories worked with a common protocol to ensure a consistency from one lab to the next. And in addition, this protocol planned to have a periodic cross-validation of test sample between laboratories. But the absolute requirement was to deliver the detection of the mutation within no more than 3 days after the sampling, which is logistically quite a challenge. But that worked quite well. That being said, the test is fairly straightforward. It's a BCR-based assay. Okay. How do I move this? Okay. So for this FLT3 assay, so this is something that today we will describe as a clinical trial assay. However, that's another aspect of that study. In discussion with the FDA, it became clear that in addition of the development of the drug itself, it was important to develop a co-diagnostic to be available at the time of the approval. And the regulatory or the technical requirement for developing a co-diagnostics are not necessarily the same as the one required to develop a clinical trial assay, even though the principle of the method is the same. So what is the impact on a study like that? The impact is that at some point there is a need to bridge the data generated within the trial with the data that will be generated with the companion diagnostic. So as an example, that had implication on the way the informed consent is written so that the patient agree that the samples are used not only to detect the mutation to determine their eligibility for the study, but also to have this sample used for the development of the code diagnostic. It also requires a centralization of all samples across the study with the aim down the road to do a rigorous bridging study that will investigate the concordance between the test used in the study and the code diagnostic. So what worked well in this collaboration? I think it's fair to say that there was a common goal from the different parties. And this is best illustrated by the very large numbers of patients that has been screened for the study, more than 3,000 in the time indicated on the slide. And 719 were actually randomized, so that's 22%. So that this percentage matches more or less with what was published in the literature about the frequency of the mutations. But in addition, and that was a concern at the beginning, but it turned out that each of the laboratories were particularly efficient in processing and returning the results of this test. Here is an example of the main lab at CALGB who had a 26 hours turnaround for the results. Now, in addition, on the day-to-day basis, there was a joint team with people from Novartis and CALGB who was responsible for the day-to-day oversight, and that worked pretty well. Now, obviously, given the complexity of a study like that, there were some challenges. And at the beginning, that required some time and some discussion to understand each other, having slightly different interests and requirement to deliver a submission may be slightly different from the perspective of improving the standard of care, even though at the end of the day, the two things are converging to the same goal. Obviously, managing the logistics on a study like that was quite complicated and required a lot of time and effort with the numbers of cooperative groups involved, with the complication of the FLT3 testing. Now, in addition, and compared to the standard of a cooperative group study, to do a registration study add additional requirements, for example, in terms of safety reporting, in terms of monitoring, in terms of auditing. And in that case, that will be the first submission for the compound. And obviously, I mentioned that already, but the development of a code diagnostic is an important component. Okay, so I mean, the study is ongoing. The result will be available in the next couple of years, but there are already some learnings emerging from this collaboration. I mean, the first is that it is important to really spend the appropriate time to keep the data flow as simple as possible. Putting together in the same study a cooperative group and industry also requires a very open and transparent collaboration between the two parties. And that sounds like easy and straightforward, but in practice, that takes some time so that the two understand each other well, that the expectations are well aligned. And in addition, there has to be some openness to use slightly different processes than we have normally, because we can't, for example, use only the Novartis SOP or only the cooperative group SOP. So there has to be a dialogue on this. And if one of the beauty of doing studies with cooperative group is to preserve the independence of the scientific and academic oversight on the study, there has to be also a balance with the operational needs in conducting the study. So in conclusion, I would think that whenever we have to do a study in a relatively rare condition where patients have to be selected molecularly, almost inevitably, that will take us to very complex collaboration involving many stakeholders, and not only here, just cooperative group and industry, but you have many other people around or many other organizations around that will participate into a study like that. So the complexity is really the sort of buzzword here. And as every clinical study, but even more with this kind of study, there has to be a range of disciplines involved that, and some of them are fairly specialized, notably on the regulatory end when it involves the development of a code diagnostic, also some technical expertise. And that's where actually cooperative groups and industry can work together, because I mean, that was on one of my first slides. I think there are very much some complementary skills here. So, I mean, this is one example. Of course, there are many other examples of industry and academic collaboration. I personally believe that this is something that has to be done more often. And one of the important consideration for me is that, particularly when thinking about rare disease, I mean, cooperative groups have an infrastructure. They are used to work together, and that makes it easier for cooperative groups to reach out to patients quickly. And that's something that we have to leverage. So with this, I will stop, and I don't know if we take questions no, at the panel. Okay, thank you.
Thank you. All right, now I'd like to introduce Dr. Hans-Georg Eichler from the European Medicines Agency. Is going to be speaking to us about the new DIGS initiative, the MIT Center for Biomedical Innovation.